Yeah, so my name is Hans Auer, and I'll be presenting my work on amygdala microstructure. So to start off, early work on amygdala function in humans has considered this region as a single unitary structure. However, qualitative examinations of post-mortem specimens performed by expert neuroanatomists have identified several small nuclei within the amygdala, each with distinct microstructural characteristics, as well as distinct connectivity profiles. These individual subnuclei have often been grouped into three larger subdivisions. As we see here, so the central medial, lateral basal, and superficial. And these three subdivisions have often been used as a template for in vivo investigations in structure, function, interaction in this region. However, we're still lacking tools to develop more personalized and quantitative maps of amygdala microstructure. And this is due to a few different challenges. First of all, the amygdala is found deep into the mesiotemporal lobe, so it's often susceptible to signal dropout. And this problem is exacerbated by higher magnetic fields. And second of all, existing in vivo approaches lack the histological validation, which remains as the gold standard to delineate these subnuclei. So this leads you to the overall goal of my project, which is to build a multi-scale framework to map the amygdala's microstructure in a purely data-driven manner. So to do this, we harnessed established features initially proposed by Axel Schleicher that's able to distinguish neocortical regions based on intrinsic differences in their laminar architecture. These variations are captured by statistical moments, specifically the mean, variance, skewness, and kurtosis of regional intensity profiles. And these features have also previously been shown to translate into in vivo imaging. For example, in this study shown here by Casey Piccola and collaborators, where she was able to use the essential moments from intensity profiles sampled from myelin-sensitive MRI. So in order to achieve this goal, we used the 100-micron resolution big brain, and we built a histological feature bank by leveraging these 4 essential moments of intensity variation across the amygdala. This was taken from the segmentations previously made in the Zhao paper from 2019, and then translated into histological space. Then these features were sampled within the essential moments were computed at 5 different kernel sizes, where voxel neighborhoods range from 500 to 2,100 microns. And this resulted in 20 distinct feature maps capturing variations in intensity distributions within the amygdala at finer and coarser scales. So to capture and visualize the underlying structure of this feature bank, we submitted it to Umap, a nonlinear dimensionality reduction technique. So this ends up showing us here the 2-dimensional scatter plot, which was reduced from the 20-dimensional feature space that we initially started with. And we showed this from the 2 axes labeled as U1 and U2, and also colored in a way to represent or to capture the access limits of these 2 components, where we see that blueness represents low U2 value, greenness, high U2 value, and redness represents the lower U1 value. Then these colors are plotted back to the amygdala space to further visualize how these components are captured. Then to further visualize which features may be driving each Umap dimension, we sorted all input features along U2 and U1. The U1 sorted matrix showed moment and kernel independent trends, where there's a correlation with high intensity values and high U1 values, whereas U2 captured more moment dependent trends. So overall, these Umap-driven visualizations of the histological feature space suggest our dimensional reduction approach could recover moment-specific as well as global intensity covariations across moments. So then to contextualize variations in this U1 and U2, in their intensities, we computed Voxelwise correlations between Umap component and amygdala coordinate spaces. So U2 was found to vary along the inferior superior axis, primarily vary across the inferior superior axis with a correlation of 0.8, whereas U1 was primarily varying across the medial lateral with a correlation of 0.3. We validate our data-driven approach to map amygdala cytoarchitecture using probability maps of amygdala microstructure. So leveraging the openly available ULIC probability maps generated from visual inspection of 10 postmortem brains, we could divide the amygdala into central medial, superficial, and lateral basal subregions by thresholding each map to show only the highest 5% probability values. So plotting these probability values, the retained voxels of each subregion, show that UMAP could dissociate these 3 established cytoarchitectural subdivisions. We find that the 3 subdivisions are primarily characterized by U2 components. This is shown this is also shown here in the ridge plots to the right, where we see where we can see the medians vary significantly. As well as U1 was able to capture a distinction between the superficial against the lateral basal and central medial. And this has to do with the subregions varying, those subregions varying across the medial lateral axis. So after establishing and validating this framework with postmortem histological data, we wanted to explore how this could be generalized to in vivo MRI data. For this purpose, we capitalized on quantitative T1 imaging collected in 6 participants at a field strength of 7 Tesla. This sequence is thought to be sensitive to cortical myeloarchitecture and could thus offer complementary insights into the maps of amygdala microstructure we derived from big brain. So these images were offered at a resolution of 500 micron, and they were run through the same framework as the big brain dataset. We used individual specific segmentations of the amygdala that were obtained with VolBrain, an automatic and openly available segmentation algorithm. And once again, a feature bank was rendered from the same central moments calculated from histological data, except the kernel sizes now varied from 1 to 5 rather than 2 to 10 in order to take in account the difference in resolutions. This again resulted in 20 distinct feature maps, which captured variations in intensity distributions at finer and coarser scales. Then I also brought this, submitted this to UMAP to get it back in the 2-dimensional space and plotted the colors back to the amygdala coordinates. And we find similar trends that were found in the big brain data. So finally, to validate this application of this approach to in vivo data, we wanted to see if spatial variations of the 2 UMAP components uncovered in each participant followed a similar pattern to what we saw in big brain. When correlating the UMAP components of each subject across the 3 coordinate axes, we find very similar neuroanatomical trends in all subjects to those found in histological space, where the U1 vector is primarily and significantly correlated to the medial lateral axis, and the U2 is primarily correlated to the inferior superior axis, as well as significantly correlated to the medial lateral axis. With these findings being consistent across all 6 subjects, there seems to be quite good generalizability with our approach from histology to in vivo in amygdala microstructure. So to conclude, we propose a theoretically grounded approach for data-driven explorations of the human amygdala, which could also apply to other subcortical regions. This approach was investigated in histology and translated to in vivo QT1 images. These methods hope to pave the way for future large-scale and in vivo investigations of amygdala organization, such as studies exploring associations between structure and function within the amygdala. I'd like to thank all collaborators, my lab mates, and as well as everyone here for your attention. Thank you.